Okay, you're an early riser, as I understand.
Yes, I am an early riser.
Okay, so we're going to start straight away. Yes, it is my pleasure to introduce Douglas, who is a researcher at the University of Arizona in Tucson in the USA. And he's the director of a large microscopy facility there called the Cellular Imaging Facility. Doug has been very interested in the ethics questions. And he has published several articles about that. And I would like to, yes, I'm looking forward to listening to you. So we will have a little lecture and then some activities afterwards, a workshop.
Okay, let me share my screen.
Please.
And it looks like everyone can see that.
Hmm.
Okay, so yes, the weather and the climate here are a little different.
Lots of
Um,
snow here.
yeah, wi- the parts of the U.S. that usually get snow are not getting snow, and parts of the U.S. that usually have winter are getting the snow that's not in the western U.S. So, but we've had a nice winter, and we're having lovely wildflowers now because our spring actually happens this month.
Okay.
So let me get started here. Working in science, we're always subject to regulations. If we do human subjects research, working with animals, biological safety, laser safety, all those things. And if we violate those regulations, there's often fines or even criminal prosecution. But in the US, when we're working with scientific images, there are two agencies that primarily do the bulk of the grant funding, and they're particularly interested in misconduct or bad behavior on scientists' parts. And the NIH in particular has this definition of research misconduct. It calls it fabrication, falsification, or plagiarism. And just to elaborate on what that is, fabrication is just making things up. Falsification is manipulating things in a way that doesn't accurately represent what happened in the research. And plagiarism is taking someone else's work and basically portraying it as your own without properly giving them credit. And this little caveat here, it doesn't include honest error, which in this case, sometimes it's hard to prove that your error was honest, um, or differences of opinion, and we'll come back to that. And I looked a little while ago, and Sweden has fairly similar language with some concerns about deviations from good research practice and also issues with negligence. And in my experience from keeping up with the literature, a lot of the violations of or research misconduct involving images involve fabrication of falsification, which is probably what you would be expecting. Um, it's taken a lot of years to come up with an understanding of what is the best practices because there are some issues with if you're in transmission electron microscopy, some things that are allowable there would not be appropriate in fluorescence microscopy. So it makes coming to a consensus across the full range of ways that digital images are used a little difficult. And I often tell my users that there's that clause in there about the differences of scientific opinion not being misconduct. And that's critical because if you tell people exactly what you did, if you give them your protocol of how you process the images, then the reviewer can think that perhaps you don't have a lot of skill or that you are completely incompetent, but you're certainly not cheating at any point along the way there. So how bad is the problem? If the name Elizabeth Bick is unfamiliar to you, she has developed a reputation in the last 10 or 15 years of being one of those people that can look at images and see strange things and patterns. And she spent quite a bit of time working with two other authors, looking at over 20,000 papers in multiple journals. And when they could pick up on changes, mostly in gel images. Uh, they agreed that they compared notes and came to an understanding. And her publication says that almost 4% of the gel images that they looked at had features suggestive of deliberate manipulation. So that's a number although it seems small, it's way too high. I looked through the case summaries from the Office of Research Integrity, which is the investigative arm for the NIH. There were 26 cases during this time period, uh, and I looked into for words that indicated image manipulation, and about 80% of them involved that. And unfortunately, one of the people was someone whose name I recognized because I trained his lab on a microscope here at the University of Arizona. I looked through the Retraction Watch database, and this is from 2023. Recently, I saw that in 2026, that database is now up to 62,000 retractions with all categorized, and about 14% of the ones there had image ethics issues. And there was a couple of big scandals in the US, one at the Dana-Farber Institute at Harvard, with several of the key leaders of the institute being accused of gross manipulations of images. And I saw just recently that there was a whistleblower lawsuit. In the US, you can make accusations of misappropriation of grant funds and get a portion of whatever award that the government gets out of the suit. So they settled for $15 million, and the whistleblower got 2.5 of that. So and the whole lawsuit settled in 20 months, and those usually take many years to settle. So clearly they had a pretty good case. And in the case of this faculty member who was a leader in the National Institute on Aging, his the accusations and the number of images that were manipulated in his research have really called into question the whole paradigm of the amyloid plaques in Alzheimer's and how the research has moved forward with drug testing and things like that. So, and I found out yesterday when I was looking up his name to see if there was a resolution to this, that he's on 800 papers. So, and that they're all going to be in that aging sort of field. Um, that's a lot of, it takes his whole life work and puts it in a real big question mark. Oops, did not want to start my web browser. So let's go over a couple of key topics that you've been talking about. You've been working on this for 2 weeks now, Sylvie?
Oh, this is the fourth week
Fourth week. No wonder
altogether.
everybody's looking tired. It's 7 o'clock in the morning for me, but it's the end of the day for you all.
And tomorrow is the last day.
Yes. So we want to think about things about improving our optics, remembering some key points about detectors and some things about digital images. I, if you look at the pictures in things like the Nikon Small World contest or some of the other microscopy photo contests, they're gorgeous. They're just beautiful images. And, when we look at, for example, photography contests, um, where you have someone who has tens of thousands of dollars worth of personal or professional grade photography gear, they can take better pictures than I can with a cell phone. But in microscopy contests, quite often people are using the exact same instruments that you have in your core facility. How come they take better pictures than we do? And honestly, the truth is that most of them clearly understand the importance of sample preparation. The sample is part of the optics of your whole instrument, and if you get that right, then the instrument will perform at its highest level. And convincing people of that is sometimes a hard sell, but it's really critical. And one of my colleagues has said 90% of good imaging happens at the bench, not at the microscope. And the detector, as you've learned in the last few weeks, is what translates the image from the analog world of optics to the digital world of working on computers. And the choices we make there are critical. What happens when we take an image that's taken on a 16-bit camera and we drop it to 8 bits? What have we lost? Not always a terrible thing, but it's a choice that needs to be thought through. When we're sampling, are we sampling to get the maximum spatial resolution, or are we sampling to get the maximum sensitivity and speed? Probably talked about that triangle of frustration where you have to choose between temporal resolution, spatial resolution, sensitivity. All those things are hard choices, but they're important ones to think through. Noise. There's, all detectors have noise at some point. Um, 16-bit camera, if you're taking a very bright image, you'll never really have to deal much with the noise. But if you're taking a very low light level image where you can't get much in the way of an exposure, you're going to have a lot more noise to deal with. So how do we ethically and scientifically deal with the fact that noise exists in our images? And whether we're saturating the image or our black levels have been manipulated such that we're hiding some of the background. Um, and just looking at the histogram, I find that it's a really underrated tool, but it tells you so much about the image, and particularly things with saturation, or if the image has been manipulated, you can often tell a great deal from the histogram. So a digital image is a process of this analog to digital conversion. It's data. It's a numerical sampling of that analog data. And it may be an imperfect sampling, but it is what we get. They are bitmaps. And to illustrate this, I took a picture from our structured illumination microscope. I stripped out one channel, zoomed in a little bit, and this is what the bitmap looks like for that channel. If you look at it carefully, the low numbers are single or double digits. That's the black background. And you can see the brighter part of the image is the higher numbers. Images typically come in a file format. Um, Oftentimes, you have a Zeiss format, a Leica format, a Nikon format. Um, and the proprietary formats contain metadata. It's kept the poor folks at Bioformats very busy trying to keep up with all the different proprietary file formats. Um, Exporting to a standard TIFF will give you an image that you can open in just about anything, but you lose a lot of metadata in the process. Information about how the information was acquired, your exposure times, your filters, all those things. And JPEG is just not recommended for image data. I know it makes for smaller images, but you worked hard to take those pictures and JPEG will smudge them. So we go from proprietary formats to more common formats, and unfortunately, somewhere along the line, we do lose that metadata. Remember that when you're working with image data, because it's a bitmap, it's an array of numbers, any manipulation of the image is, in a sense, a mathematical function that's being applied to that array of numbers. I've never met an accountant that would apply a mystery function to a spreadsheet so that it looked better. So we should be careful about applying functions to our images as well. And it's sometimes really hard to tell what sort of post-processing has happened to an image. This is where you need to keep track of what you've done. Some manipulations are considered fairly benign, like a histogram stretch, but others are changing the data in a way that does need to be reported. And frankly, a lot of people just treat software as a black box. It's like, well, I just put it in there and stuff happened. Well, we need to know at least a little bit about what's going on in there. I wanted to throw a new slide in this year about AI and imaging. I've been paying attention to this, but this is also moving so fast. I've been asked several times, should you write a paper on AI and image ethics? And it feels to me like trying to hit a target while riding a galloping horse. AI is changing so fast, um, but it's a useful tool in the right context. For example, in image analysis pipelines, AI segmentation can do things that in the days of just doing thresholding, we could never fully separate things. And there's a lot that's worrisome about AI because oftentimes the vendors oversell it. Uh, all you have to do is read the news, and have people are saying this AI this is amazing, and then 3 months later they come out with a new model which is so much better than the last one. Um, and there's rarely any caveats that AI has hallucinations, makes stuff up, and all that kind of things. So it's really critical, no matter how you use AI, to have a knowledgeable human in the loop so that you're checking the work of the AI so that um, that you're really getting legitimately good answers to the questions that you're asking. And frankly, the capabilities of the whole generative AI subset of this are quite frightening. And to illustrate that, um, I did not see these originally myself. I got them from a colleague at the University of Massachusetts. They were on the Blue Sky social network. These are generated from a text prompt. And frankly, they're pretty convincing. So we are, and we have unfortunately, in the Tucson area, there's a national news media's family member who was kidnapped in the Tucson area, and they're concerned about generative AI creating, doing the voice, the deep fake of the voices. It's like, yes, is this person's mom okay? Well, all it takes is a few sentences, and AI can do deep fakes of voices now. So it's going to be a real issue going forward. All right. Hopefully, I haven't talked too long and we can get on to the discussion. Uh,
Yes, that would be nice. So are you